I'm sorry about this. Just a moment. I'll just connect it. Uh, okay. So, yeah. So, as we are looking at these deep learning models, explainability is one of the major point where uh, I see why my model is able to predict whatever it is predicting. So, what is the reason behind that? The main thing that came into focus is I am able to correlate whatever my model is predicting. You can see the normal. So, this is a normal heart, and this is the myocardial infarcted heart. You can see there are certain tissues that are actually having some ischemic. We call it as ischemic condition where there is some the blood flow or the coronary artery is having some blockage, and that leads to a blood flow obstruction that leads to ischemia, myocardial infarction, and so on and so forth. So it's a chain of events. Okay, whatever is happening is a chain of events. So I want to identify that. So if you can see the normal signal, how it is looking, and the myocardial infarcted signal, how it's looking, you can, it is very, very difficult to continuously monitor it and tell a very accurate interpretation of whether it is, your heart is going into a infarcted mode or not. Okay, so our model is able to capture that. And we are also able to see it with the literature, compare it with the literature, and see why it is doing so, why it is able to capture it. So you can see it is actually looking at certain. So this is the explainability part. So what is how are we going to interpret this explainability wherever the white regions are there? So that is or the bright region is there. So that is where the model is actually looking at. The other regions are not being captured. Okay. So if you can see in this particular wave, in one way, I'm talking only about this wave or this particular one beat. Okay, so here, the white portions, if you can see, it is mainly concentrated on the ST segment and a part of R wave. Okay, part of R wave and ST segment. That's all. So, which is well in alignment with the literature, where in during myocardial infarction, what happens is there will be something called to be as an ST elevation. You can see this is the ST segment. This is S and this is T, where ST segment between this S and T wave, I will have an isoelectric line. Isoelectric line means a flat line I will have. In myocardial infarction, that flat line will not be there. Rather, you will have a slant in the an elevated line will be there. So that is called to be as myocardial. That can be diagnosed as a myocardial infarction. But now this using, I mean, a doctor screening it time and again will be very difficult. So your deep learning model, which can be integrated with a wearable, can do this. So any developed model, it can be deployed on a simple controller. Okay, based on how heavy your model is, it may require Raspberry Pi or something that is a heavy, or maybe Jetson Nano or something that is a heavy processing, which has a high processing power. But these these controllers can be deployed, can be utilized to deploy the model, and we can be able to predict whether a person is going into a heart failure stage or not. So that is how this can be utilized. Okay, well and good. We have seen about wearables. We have seen about intelligence, how intelligence can be integrated into wearables. The next hot topic in wearables is IoT. So why IoT? As I told you previously, I want to connect multiple things and I want to transfer it with respect to a communication platform. Also, I want to get suggestions from the doctor on how my body is performing. So all these things are important. So and one more thing is, I may have multiple things in my body. You can see I have phone, I have Fitbit. Right now itself, you can see I have a Fitbit, I have phones, I have I can have a smart shirt that I can wear. Smart rings are there, smart fingers. So there are multiple wearable devices that a person can wear at one time. So if you can connect all these things, the information that is gathered is more, and the diagnosis that you can give using all these wearable devices is um, uh, much more convincing and much more effective, rather than individually analyzing the data. So IoT came into picture. So what is this IoT? I connect all my wearables into a common platform, and that common platform I will utilize it to utilize it for analysis and some prediction and some detection of abnormalities. Okay, so how is it carried out? So mainly, okay, prior to this, so where we generally use it, specifically during the era of COVID, you can see a very high uh, the very high need for these kind of technologies, that is telemedicine and other things. So you can see any data that is being sent, health data is there. So you have a health data that is actually sent to a doctor as for diagnosis, or you can go in for any data display utilities, or you can go in for different smartphone based technologies that can control, that can tell the patient, okay, you are having these kind of problems, so kindly take care or kindly go to a doctor. So these kind of technologies have been developed wherein you need a communication pathway, you need to connect the different things, and you need a communication pathway to establish these connections. Now there are many devices that are approved in today's market. So FDA approved devices. We have telehealth monitoring systems. We have different PKG watches. We have these kind of diabetic insoles that are available. So this can be utilized for monitoring the pressure during different gait phases. Okay, gait, whenever we walk, the posture is different due to, and that differences in posture can lead to different I mean that can talk about different abnormalities. So all these things can be monitored. And we have ambulance monitors, we have different kinds of epileptic seizure measuring devices. You know, seizures are very important to be diagnosed at a correct time, because when a seizure is left unattended, there are problems that can happen to the brain of that particular subject. So these epileptic seizures, there are many people who have died due to these epileptic seizures. So there is a very high need for these kind of devices that can monitor the presence of seizure, or that can predict, instead of monitoring, I would say predict, that can predict. So in that area, people are still working in, where I want some technology to predict, okay, fine, this person is going to have a seizure after this amount of time. Okay, or else if I can predict within 5 minutes, okay, this person is going to have a seizure, then I can control the seizure from happening. So we need these kind of devices that are available. So right now seizure monitoring devices are there. Right now heart monitoring devices and other things are there as FDA approved devices that is available in wearable IoT market. Okay. Now, how is the market? Obviously, if I have a very good connectivity, if I have a very good wearable technology, I have a very good AI based technology, then I will have a very nice IoT based technology in the market. So you can see the global percentage growth is very high. Also, Indian market is also playing an important role. But where do we need these kind of telemedicine based, IoT based telemedicines? Mainly in for different kinds of applications. We have seen a very high uh, improvement in neurology, teleconsultation, telemedicine, teleeducation also. So there are many things, but you can see a major chunk of it is occupied by telemedicine, okay, and teleconsultation. So that is how you can see that if you're doing a research on IoT based devices, it is a you can quickly commercialize it, and you can make it into the market, and you can sell those devices to create an impact on the society, or to have a personal career growth. So all these things is possible. So always having a very good market growth in that particular era, in that particular area, and working in that area to give a good outcome. So that will always play an important role. Now, what are the advantages? We know very well. So if I have a telemedicine technology, then I have better access to the healthcare. It provides a cost effective nature, because I don't have to spend time to go visit a doctor and do things. It is convenient. It doesn't depend about whether the doctor is available or not. It just, you have an artificial intelligence technology along with along with IoT, that can integrate these two platforms and give you a better prediction using these kind of using the data that is available. Okay, so what did we do in that? this is our lab work that we are so I currently I am we have seen till okay device development, electrode development that we have carried out along with our collaborators. We have done this electrode development, and we are doing AI integration with that electrode development, so that has become a device as such. Now, how to integrate it with the server or with the Bluetooth, the wireless device, so that it can be utilized for monitoring. So right now, what we do is we are generating these. We are patient having these ECG signals are being captured by using a Bluetooth device. So right now we are using it using mobiles to capture these device, and whenever we capture these data, your mobile also has a limited availability of space. So what we do is we directly upload it into the AWS cloud, and we use cloud services to because these cloud services can be used to deploy an algorithm, so deploy any deep learning model or machine learning model or whatever. So and it will give you, based on the data that is uploaded, it will give you a prediction, and that will keep on monitoring whether whenever the prediction crosses a threshold, that is whenever you get an abnormal data, more abnormal data than a threshold limit, then it is indicated to the patient that there is some abnormality that is going on, you may please visit the doctor. Okay, so these kind of technologies have been developed, and so what is the advantage? We have a 24/7 monitoring. We have different kinds of local data saving techniques that are available. You can view the data. We can have a graphic display, and we can also have heart rates that can be monitored on the mobile phone. Okay, so you wear this, you can monitor the heart rate on your mobile phone continuously. Right now, you cannot do that in your mobile. With your digital watch, it is not possible to completely monitor your cardiac data continuously using your digital device. But right now, with our technology, you will be able to do it continuously with your mobile whenever you are, and it will be stored continuously. You can take a graph of it, you can do whatever analysis you want, or you can see how your heart is behaving, other things, whatever you want, you can do. So right now I'll just show you, this is how we have done it, and this is a basic graph. Don't so you can see that this is how your data will be recorded and it will be shown. So you will get a very nice ECG signal rather, and you will also get an instantaneous heart rate that is displayed. So heart rate will also be calculated continuously and it will be displayed. Now, okay. So well and good. So we have seen three things. One is about different cardiac monitoring devices that are available in the market, how we can integrate AI technology, how we can integrate IoT with it, and create a complete device that can be utilized for monitoring cardiac potentials. I am going to slightly deviate from that. So, what are the different wearables that are available for different purposes? So, let us focus on another parameter that is called as electrodermal activity. So, this is another signal that your body sends in. So, we are not. This is kind of an induced signal wherein I am going to send a small current, a very minute current. You will not even feel that current actually. A small voltage is given across your finger. So this is a device that is there. Currently, this is actually a research-based device. Currently, these devices can be integrated along with your watches also. Okay, so this is a device that is available to record the skin resistance changes. Okay, so why this is important? So whenever a person undergoes some kind of emotional change, okay, there are multiple consequences that are there in the body. Meaning, I am very happy. My body sends in different signals stating that I am very happy. So, what are the things that are essential? I can capture it using a camera. I can analyze my face, and I can say whether I am happy or not. Well and good. But these facial techniques, whatever we are doing, are not going to give you the correct information, because it can be forged, right? I can, even though if very happy, I can also make a sad face, neutral face, and I can continue with my work. So your video based technology is not going to help. So we are looking out for alternate technologies that can understand the differences in the body that is happening. So one among them is electrodermal activity. So this is used mainly for this is mainly used for something called to be as lie detector. If you know these lie detectors that they use whenever there is an inquiry that is happening, you can see they will connect. So whenever a person says a lie, so they say that it is an evident fact. So the resistance of the skin changes. Okay, so that is how they use this lie detectors. That is also one technology that is used, but we are going to use it for another purpose. So what purpose we are going to use it is like kind of an mind awareness. So whether I'm concentrating or not, how do you know I'm taking a class, or many of you will be many of you are faculties, I think. So you are taking a class now. So when you are taking class, how do you know whether the person is concentrating or not? So it is impossible to identify whether he is really concentrating or not. So for that, there are different um, technologies that have come up, video based technology, whatever I'm saying, but that will fail because anything on the face can be forged. So we are going for another technology called to be as mind awareness using electrodermal activity, wherein this is an involuntary signal, or you can say we are body is not controlling it, or we cannot control the signal from happening. Okay, so the physiological response happens due to the variation in the mind awareness or mind alertfulness. Now, basically, any electrodermal activity signals, or EDA as I call, it will be taken across time. So we had done a particular methodology that can be utilized in terms of using EDA for understanding the mind activity. Okay, so we have taken EDA signals, and we have done a pre-processing technique. So pre-processing as in to remove any noise that is present in the signal, because whenever we are recording a signal for monitoring any physiological activity, it will be associated with noise that are inherent due to environmental factors, due to electromagnetic interference. There are many things. Motion artifacts will be there. So there are many things. So based on this, you need to employ an algorithm that can be used to remove noise. So noise removal we have done, and we have followed a particular technique called to be as empirical mode decomposition. So this, what it does is it will decompose the signal into multiple functions or multiple mode functions that have multiple frequencies. You can see this is the signal, this is the actual EDA signal means it is divided into multiple frequencies. You can see these are multiple signals that are derived from the original signal. Okay, so why are we interested in this? Because as per the literature, we want to understand how what frequency is mainly important, what frequency is mainly important in understanding the mind alertness. So we are going in for this kind of approach, and you can see these are the two signals that are coming from EDA signal recording. So this is a mind aware signal, and this is a absence of mind awareness. Okay, so based on signals, you will not be able to identify anything, nothing. So you will not be able to say whether using this EDA signal, you will not be able to see, okay, this person is having a this person is concentrating or this person is not concentrating. So we need to analyze, we need to understand the key characteristics of these signals. So when I decompose it into multiple mode functions or intrinsic mode functions as we call, or multiple frequencies, you can see how it is behaving. So for different frequencies, you will get different kinds of mode functions, and we try to extract some features from these mode functions called to be as entropy measures. So how random are these mode functions? Okay, so if I consider different mode functions, this is IMF 0, 1, 2, and I can take how many ever mode functions I want. I can divide it into how many ever is required. Right now we have considered 3, and I am going to take I'm going to do an analysis on. So this is the pipeline that we have followed. So similar pipeline is available for multiple techniques. So I am just giving you an example on how you can do an analysis based on any variable signal, and you can draw conclusions from it. Okay, so you can as we can see that we have calculated entropy measures, which are we had taken three entropy measures, approximate entropy, sample, and SVD based entropy measures. And we analyzed what IMF plays an important role, which IMF plays an important role in these entropy. So in differentiating these signals. So you can see I am giving a percentage deviation between mindfulness and mindlessness, or aware state and no aware state. Okay. So you can see the deviation is very high for IMF2. Okay, when we talk about approximate entropy and SVD, when we consider sample entropy, your deviation, I mean deviation is higher for IMF0. So when we think about these measures, we are interested in identifying a particular signal that can be utilized for further stages. Why? Because see, you cannot process the entire signal as such. The problem happens in, see, you need to process everything in real time. You should have a adequate computational power for processing that. So if you design a methodology in such a way that I am not going to utilize the entire signal, I am going to use only a part of the signal to understand whether that part plays an important role in differentiating between mindfulness and mindlessness, then that is a key significant factor in designing your wearable based intelligent based wearable devices. Okay, so now what we found out was IMF2 is very good. Okay, so the frequency measures that correspond to IMF2 can be utilized for differentiating between the mindfulness and mindlessness. So that we have also tried to prove it using hypothesis-based statistical analysis. Okay, so we got a very good statistical measure or p-value for the IMF2 for all the entropy values, but among them, approximate and SVD plays is good enough to differentiate between these two classes. Okay, so okay, this is the third thing that I wanted to touch upon, because this is also one more thing that we are working on. How to use any wearable devices for understanding cognitive impairment. So, just a basic idea about what is cognitive impairment. Cognition refers to brain activity. So like for example, I am thinking, I'm having a perception, I'm reasoning something. So all this comes under cognition. So these are all brain activities that are essential for everyday functioning, so our day-to-day activities and everything. So whenever there is an impairment that happens in cognition, it involves some kind of memory issues, when there's some kinds of deficits in thinking process, then we call them to be as cognitive impairment. Okay, so this cognitive impairment, actually, it is a very crucial scenario wherein many people in the world are affected by this cognitive impairment. If you see elders in our country, there are many people. The aging population is quite high actually right now. So there are many people that who are actually having cognitive impairment, but they are not diagnosed properly. Why? Because in a country like India, as I told you, we have a very poor doctor to patient ratio. And because of this, not every doctor is adequately involved in understanding the situation of the geriatric population. This cognitive impairment mainly affects geriatric population, that is age-old population. And if you see globally also, approximately 13% are affected by this disease. Now, when we look into normal aging, like as we age, okay, a certain degree of cognitive impairment will happen. But so if we think about our elders, they will say, I am continuously forgetting some things, right? So that is a certain degree of impairment that is happening in our brain. But there are certain disease conditions that enhance these degradations, meaning there are degenerative disorders that we call that leads to cognitive impairment, where the degree of impairment, that is the amount of impairment that is happening, is many folds. I don't know if you had seen, many movies have depicted these kind of disorders where a person tends to forget his or her own name and address nothing. They cannot remember themselves, so it is very difficult for them to survive. So okay, what can be done for this? The current technology utilizes something called to be as MRIs. MRI are the structural magnetic resonance imaging. So you take an MRI, you try to diagnose whether the patient is having a degenerative disorder or not. So this is a normal MRI and this is an Alzheimer's disease condition wherein significant changes are seen between the normal case and the Alzheimer's disease case. You can see this is enlarged, right? So this is a ventricle that is present in the brain, a fluid-filled cavity. This is enlarged. You can see it is smaller here, right? So that is how the progression that is how the progression is. So most of the brain tissues are dead by the progression of this neurodegenerative disease. So if I can diagnose this in a very earlier stage, like for example, it starts. So this particular neurodegenerative disorders, they take time to progress. Like you take a span of 10 years to progress from a normal aging to a diseased condition. Okay, so it takes 10 years to progress. So if I can diagnose it in a very earlier stage, like before it progresses towards Alzheimer's condition, then or any neurodegeneration. Right now, I have considered Alzheimer's. So there are many kinds of neurodegenerative disorders. So if I can identify it at an earlier stage, then it is very easy to stop the progression of the disease, or I can say to delay the progression. So if life expectancy of the person after disease diagnosis is 3 years, if I can diagnose it at an earlier stage, I can enhance the life expectancy. So there are treatments that are available that can enhance the expectancy of that particular patient. So like this, we can actually control the progression of the disease. Now, an average person, you can see in a population like India, I will not be using I will not be taking MRI time and again. Not possible, right? So why are I am normal. I mean, a subject who's feeling normal at his at about 50 to 60 years of age. Why will he go and take an MRI? Because at this point only this early mild cognitive impairment starts, the neurodegeneration starts. Okay, so he may not be aware that he has to take an MRI to identify whether there is a condition that is happening with his brain. So we need some devices that can actually identify these early mild cognitive impairment stage without even using MRI. So what we tried was an alternative route. So can we use speech signals to diagnose these Alzheimer's disease condition? So why these speech signals? Because brain, whenever it undergoes any changes, but specifically during the early mild cognitive impairment stage, what happens is a patient exhibits difficulty with speech. He may start to stammer. He may start to bring in some unwanted tones. Okay, there will be changes in his pitch. There will be changes in some kind of difficulties in familiar task completion. So all these things are also exhibited as symptoms by the patient. Okay, so why can't we use this difficulty with speech and try to find out a wearable device that can be integrated with an AI model, and it can be utilized for continuous monitoring of the speech signals to identify whether the person is going towards any abnormal conditions. So what we tried is we took a few samples of audio samples of speech samples. These are all speech samples, but these are all speech acoustics, meaning I am not interested in how a person is framing the words. I am interested in more about tones. What kind of patterns are there in his tone, and what is his pitch, how variations in pitch are there. So that is what I am interested in. So we took the acoustics of the speech signal, so for a control case, and this is for a dementic case, and we try to pre-process it, because there are multiple environmental noises, surrounding noises will be there, and other things. So there are multiple speakers who may be speaking at the same time. So I want to take only the patient's speech. So you can do something called to be a speaker diarization, that can be easily integrated with your controller. So it is a small a few chunks of code that can be used to automatically diatrize whether the speaker that is of interest is only being monitored. Okay, and after that we can go in for something called to be as feature extraction. I had told you these things. So now, what we had calculated is something called to be as time frequency analysis or spectrogram based analysis. It is basically doing a frequency domain analysis, meaning I am interested in how the frequency changes are between a normal subject and a dementic subject. So this is a spectrogram representations of a normal subject, and these are all for abnormal or AD subjects. Now, just looking at it, we will not be able to gather any ideas. So we try to understand how the variations are in the substral coefficients. So we extracted something called to be a substral coefficients, and we try to plot those substral coefficients in differentiation. You can see the there are these are the statistical measures that are used for differentiation. So I can see mean, median, mode, all but parameters are there that show differences. Some parameters show very good differences, some parameters were not able to differentiate well. So we are actually working on this, still project is going on this, and we are trying to make a standalone device that can be used as a just with the help of a microphone and a controller, a small device that can be used to monitor the speech signal, monitor the tonal capabilities of that speech signal, that can be used for understanding the understanding whether the dynamics, speech dynamics of the speaker. Okay, so this is a model that we had done. So this is a machine learning model that we had implemented that is based on random forest, logistic regression, and multi-layer processor. These are all models that are available that can be trained on your training data set, and it can be tested on a new data. And we had got about 75 to 80% accuracy. Okay, so in differentiating between differentiating between the normal and the dementic subjects. Now we have considered it as a standalone pipeline, but this can be integrated along with the wearable, and it can be utilized for understanding the speech dynamics of the or acoustic speech acoustics of the subject. So this using this okay, so using this we are able to understand there are multiple variables that are there in the market for different purposes. There are multiple technologies that are there that can be utilized for different purposes, and I want to integrate these technologies, and we can bring out a very good future when we can integrate these technologies, and when we can utilize them for betterment of the society and betterment of our healthcare. So with this, I will just stop here. So these are the different works that we are doing. I have just given a few samples of whatever is being done in our lab. I have not gone into depth also, so because it's a quite a short session to go into depth in multiple things. So we work on different kinds of image processing, pattern recognition projects. We also work on creating different machine learning and deep learning pipelines, algorithms, and we also work on electro technology in collaboration with different people, and we also work on multiple image guided intervention. So if you are interested in any kinds of collaboration and any uh, uh, any kind of collaborative projects also, so you're welcome. Please mail me. This is my email ID. So rohinip at iiddm at .ac.in. You can mail me. And if you're also looking for PhD scholars who are interested in doing their PhD here along with our lab, so you can, if any potential candidates are there, even in the talk or your known circle, you may please ask them to mail me their resume, and we can probably take it from there. And we have a project opening also. So it is based on developing deep learning models. So if anybody who is interested, they can send their resume to my mail ID. And yeah, so that's it from my end. Thank you very much. And if you have any questions, you can please ask. I can try to answer whatever is your doubt.
Dear participants, if you have any queries, please ask Dr. Rohini ma'am. If not, we can wind up the session with a thanking note of Rohini ma'am. Uh, I'm
Yes.
asking once again, is there any questionnaires? Questionnaires session is open now.
Excuse me, ma'am.
Yeah.
Ma'am, actually, is it possible to identify lung cancer using any wearable device?
Yes, actually, right now projects are going on with respect to sound-based information. So we had a collaborative project with respect to Amrita College here. So they are working on lung sounds identification and differentiating it for finding normal and abnormal, finding different kinds of abnormalities. Particularly, if we can see lung cancer is associated with multiple scenarios where the progression starts from an earlier stage to the terminal stage. And whenever there is an abnormality that is happening, that is growing in the lungs, there are scenarios where the lung sounds are potentially different. So it is possible. There are no FDA approved devices as of now. It is still in the research stage. So papers are there which take this into account to get a clarity between what features to extract, how to differentiate. Even deep learning models are there. So, but currently there are no wearable devices that are available in the market as such.
Okay, using sensor, we can
Yeah.
they can using inhaling and exhaling capacity, they can identify the abnormality, right, ma'am?
Inhaling and exhaling capacity requires some kind of setup that is not very convenient for wearable, right? So if we can think about anything like heart sound, sorry, like we are measuring heart sounds. So there are technologies that are similar to measure something called to be as lung sounds. Lungs exhibit, whenever we are inhaling and exhaling, there are certain sounds that are produced by our lungs which can be captured effectively. So these can be utilized further for analysis.
Is it related to COVID-19, ma'am, that lungs capacity like this?
Pardon, COVID-19 in the sense like we do have variation. So thinking about lung as such, there are variations that are presented by the lungs in terms of lungs are also contracting and expanding. So whenever there is a motion that is happening, there are certain sounds, certain vibrations that are produced, and these vibrations can be directly related to abnormalities because the pattern will be different. So identifying these patterns are only important. So we are going to identify these patterns using some kind of intelligent technique, like some AI based technique. So that AI based technique that we need to identify, that is similar to whatever we are interested in. So COVID-19 also presents some abnormalities, abnormal sounds. So based on different scenarios, different abnormal sounds will be exhibited. So if I can think about a small example that can be connected, you can think that we know that heart sounds are there are different heart sounds that are possible, right? We have what is that? Whenever there is any valve abnormalities that are there, I will hear a small murmur. That is how the doctors identify the valve abnormalities. So these abnormalities can be picked up by something called as phonocardiogram. So some sound based wearable device. So similarly, lung sound can also be measured, and that can be utilized for further diagnosis.
Thank you so much, madam. It's
Yeah.
very informative. If it's possible, kindly share your PPT. That's really useful for us.
Hello, madam.
Yeah, sure.
Hello.
Yes.
Madam, we can use all persons, madam. Any limitations are there?
Sorry?
We can use any person or any other, some rules, regulations are there?
Yeah, we do have. Whenever we want to use, see, even for the basic research that we are going to do, like for example, if you are going to do a non-invasive measurement, like you want to measure ECG signal, we need to get ethical approval, ethical clearance, even for research stage. So whenever we are going to bring it in the market, even for a non-invasive device, it is needed. There are certain regulatory measures that are required for it to be compatible with. So it shouldn't cause any harm. We are going to measure on skin, so it should be biocompatible. It shouldn't have any reactions to the skin. So all these things are needed. So we need to follow certain structures. Some approvals are required. So that is all in the next stage of research. Like for example, if you're going for clinical validations, there you require FDA approvals and other things so that you can go in for clinical studies.
And, second thing, madam, in YouTube, generally frequently we are getting number of devices, whether it is
Yes.
believe or not.
See, there are many devices in the market. So digital watches have, they say that they can monitor stress levels, they can monitor heart rate continuously, all those things. See, data dependency is always a question. We don't know how accurate it is until and unless it is actually having a proven record. So always measuring it with respect to sensitivity. So that is what I had already told you. Sensitivity is very important whenever we talk about developing any device. So how sensitive it is, how repeatability is there. So all these things are needed. So if it is clinically evaluated and clinically tested and proven, well and good, it is good enough. There are multiple devices in the market which are not even clinically approved. So we have to use our discretion to identify whether we need to do some background research, whether it is good or not before utilizing it.
Okay, okay. Thank you, madam.
Yeah, thank you.
Ma'am, I have one question.
Yeah. Please.
So you showed about the explainable AI that you have used for myocardial infarction detection.
Right. Yeah.
So can I know what explainable AI did you use?
So we use a basic GradCam.
Okay.
So GradCam can identify how the feature maps are.
Mm.
So where the alterations in the feature, which particular, so if I consider, if you think about network or neural network, so it gives you different stages. There are different stages in a neural network. So at every stage, multiple levels of features are being extracted. For example, we call it to be as high level features, low level, mid level features. So in these features, among the different features that are extracted, which is actually giving you, contributing towards that output. So that will be given by a GradCam map. So that is actually plotted and shown there in my PPT.
Okay. Okay. Thank you so much, ma'am.
Thank you.
Okay, ma'am, I have a question.
Yes.
From a healthcare sector point of view, suppose there is a patient who is wearing multiple wearables.
Yeah.
So, in near future, to detect all the signals on a common platform, will they not interfere with each other to come to a actual authentic result? Like a pacemaker, when we are using a pacemaker, which is either it can be non-invasive or invasive, but there are some restrictions.
Yes.
So for wearing multiple wearables and interpreting, for example, in case of telemedicines, interpreting it in a common platform, will it be really authentic, or there may be some error in that too?
Okay, so to answer your question, there are multiple aspects actually. So there is something called to be as BAM, body area network. So it is a very recent, not say very recent, but in the early 20s it came, like 2022. It has been in the very hot research area where we are trying to connect multiple sensor networks and utilize it for a common abnormality prediction pattern. So what happens here is there are few things that people are working on, basically in the form of communication protocols. So which is priority? Do I want information from the pacemaker to be as priority, or do I want ECG as a priority, or do I want temperature as priority? So which one is priority? Even among sensors, we can classify it as multiple. We can give priorities for multiple sensors. So that is possible. So people are working on different protocols that can fix these priorities. For example, I want what is the information from a pacemaker to be as a highest priority when compared to temperature measurements, because that is relatively stable. So we do not have temperature variations happening over minute by minute. So that is relatively stable. So I don't want that information throughout. Even if that information is not there, I will be able to predict some values that is near to it. So what is of ultimate priority, and designing a communication protocol that can capture this that can prioritize and that can refresh this information again and again. So that is being done. So with the help of that, we can create a very good network of sensors. That is one aspect. The second aspect is using a model. If we think there are technologies right now which uses multimodal approaches. Multimodal means we take information from multiple scenarios and we integrate it into one single model. I'm not using multiple models for that. I'm using one model that takes an input from multiple sensors. Okay, so and it can process it over real time and it can give me a prediction. So these are the two aspects that are there. So if you think about authenticity and priority, I will say priority plays a major important role. So communication protocols are being developed, and in the other area, we also have something called to be as authenticity. So there can be lags, there can be information mismatches that are happening, but right now technology is developing. We have multiple systems that can take in information very quickly and process it quickly. So that is also there. So yeah, so these are the different aspects where still work is going on, still integration is going on.
Thank you, ma'am.
Thank you.
Is there any other questions? The session is really interactive, actually. We are really happy that so many questions are posted from the participants, and we would like to know if any other questions are yet to ask from the participants.
Even if you want any help from my end, you can always mail me. You can contact me through my mail ID. So I'm open for any collaborations also. Please do send me a mail if required.
We do share the information of ma'am in the WhatsApp group as well as in mail.
Yeah.
So if you have any queries, please contact Dr. Rohini. She will be available at any time. I hope so.
Yeah, sure.
And with this note, I just wanted to express my heartfelt gratitude for the enlightening session of wearable technology, and your deep insights and real world applications and the topics both are fascinating and highly informative, ma'am. And your sessions are not only broadened about the topic which I've presented, but is also it has really inspired us to explore the vast potential of wearable innovations. Thank you so much,
Thank you.
ma'am, for sharing your expertise and knowledge with us. And looking forward for the same in the future also, ma'am.
Yeah,
Thank you so much for the
thank you.
wonderful session,
I'd like to
Rohini,
thank
ma'am.
Ms. Nitya for having me here. Thank you very much for calling me. Yeah,
Thank you.
thank you.
Thank you so much, ma'am. Thank you so much, ma'am. We'll wind up the session. Thank you so much, ma'am.
Bye.
Thank you. Thank you. Dear participants, I hope the second session, resource person, is also available in the session now. We'll have a short break for 2 minutes and we'll resume the session after 2 minutes, okay? Participants are requested to fill the first session feedback link as soon as possible. And we have Mr. Marimuthu sir is online and ready to present the session 2 of day 1. I request Mr. Jaiseelan sir to introduce Mr. Marimuthu sir before signing into the session.
Good evening, everyone. It is a privilege to introduce Mr. C. Marimuthu, a distinguished expert in radiology and medical imaging with over 31 years of experience, including 9 years in academia. He currently serves as the selection gate radiographer and academic coordinator at the Institute of Radiodiagnosis, Government Kilpak Medical College Hospital, Chennai. His educational journey is both diverse and inspiring, holding multiple professional and academic qualifications. He is a certified radiologist assistant and has earned BSc in Medical Imaging Technology from Manipal University, a Master of Radiation Technology from NIMS Rajasthan, and an MBA in Hospital Management from Allahabad University. His expertise in radiation safety is backed by specialized training from Baba Atomic Research Center, Mumbai. Beyond his professional roles, Mr. Marimuthu is a renowned academician and leader. He has delivered over 60 lectures at various national and international conferences and served as a scientific secretary and organizing secretary for medical events and won numerous best scientific papers and quiz competition awards and national and international competitions. His contributions extend to medical education as an external examiner for prestigious institutions like the Tamil Nadu Dr. MGR Medical University, SRM Institute of Science and Technology, and Savitha Medical College. He is also an author, having written Short and Simple Guide to Radiography Technology and KMC Manual on Radiography Positioning, which are invaluable resources in the field of medical imaging. His exceptional contributions have been recognized with multiple Best Employee Awards and the COVID Warrior Award for his dedication during the pandemic. Today, we are honored to have him share his expertise on radiation personal monitoring devices, a crucial topic in ensuring safety and efficiency in radiological procedures. Let us all extend a warm welcome to Mr. C. Marimuthu sir. Sir, you can start the session now.
Yes, sir. Yes, sir. Yes, sir. Thank you, sir, Dr. Jaiseelan, sir, for the wonderful introduction. And good evening, one and all gathered here. At the outset, I would like to express my gratitude to the management of the ATAL Institute of Technology and the faculty members of the Electronics and Communication Department. And my special thanks to Mrs. Nitya ma'am and Mr. Jaiseelan sir for their kind invitation for this faculty development program. It's a great honor to be here.
Thank you so much, sir. We're also very delightful to have you in the session, sir.
Thank you. So X-ray is discovered in 1895 and have become a vital part of the medical diagnostic and helping us diagnosing diseases, fractures, deformities. And this while they offer a great benefits, excess radiation exposure can be harmful. And with the growing use of the X-rays in medicine, ensuring radiation safety has become more important than ever. Today, I will be talking about the evaluation of the personal radiation monitoring devices and how it is started and what is happening now in India and what is the future ahead. Let us see some basic physics. What is radiation? Radiation is the energy that travels in the form of waves and particles. It further classified into the ionizing radiation, which means it has the energy to remove a tightly bounded electrons, and this is called the ionization. And that X-rays and gamma rays and other alpha beta particles comes under the category. It is commonly used in the medical imaging and cancer treatment and industrial applications, but it requires a careful handling due to potential health risk. Another category is a non-ionizing radiation. It does not have much energy to ionize the atom, but it can cause heating or other molecular changes. And these microwaves, infrared, visible lights, and ultraviolets are come in this category. This is the electromagnetic spectrum. You can see it is all the radiations are arranged based on these frequencies or wavelength. And what you can see in the blue color, it's a non-ionizing radiations. In the microwaves, radio frequency are come under this category. These are the high-low energy X-rays, low energy radiations, and these X-rays and gamma rays all come under the category of ionizing radiation, and they have the potential energy to make damage to the living cells and damage to the genetic material like DNAs. Fortunately, the cells in our bodies are extremely efficient at repairing this damage, but when it goes beyond the limit, it is dangerous. So ionizing radiation can be it's alpha, beta, gamma, and neutron particle and X-ray also. These are the ionizing radiations. So alpha particle radiation is basically it's a helium nuclei, and it is very heavy. So it cannot be penetrate, cannot travel very long, and it can easily can be stopped with the paper. And beta particle is it's a stream of the positron or electrons. It can travel little better than this alpha particle, and it can be stopped by the aluminum or small amount of the thickness of the water itself. And that's gamma. It has some higher energy than alpha and beta and even X-rays. It has higher in penetration capability. It can travel through up and it can be stopped up to the lead levels. So lead can be used to stop the protection for the gamma rays, and the X-rays falls on between the beta and gamma rays. That also can be stopped. It can penetrate aluminum and it can be stopped with the lead. To at least 2 mm of lead is more than enough to stop the radiation, X-ray radiations. But in case of the gamma radiation, we need some higher thickness of the leads because it's higher energy. And the neutron, it is a part of the nucleus. It having a higher penetration energy, and it can travel. It can penetrate the lead also, but it can be stopped with the with the substance of which hydrogen, water, or paraffin or concrete. We are concerned about this, and where it comes from. Actually, that's the source of the radiation. So the majority of the 84% of the radiation comes from the natural background, and from the that means from the ground, soils, water, and even from our bodies. And background radiation can also comes from the outer spaces from the sun. That is called the cosmic rays. Other sources are man-made, and it is 16 percentages contribute this, in which 15.5 percentages from the medical field, what we are involved in it, we are concerned about this. So some few 0.2 percentages comes from the nuclear fallout, which is the residual radioactive material propelled into the upper atmosphere following a nuclear blast. So whether we are working in a radiation field or not, that is not a matter. That you will be radiated with the 84% of the radiation. That will be around it will measured by the millisievert unit, and around 2 to 3 millisieverts are the background radiation, natural background radiation, everyone radiated every year. So radiology, what is radiology? Radiology is branches of medicine that use medical imaging techniques to diagnose and treat the disease, and it involves various imaging modalities such as I'm talking about the diagnostic radiology. So basic conventional X-ray, digital X-rays, computer X-rays are there, and computer tomography scanner, mammogram to diagnose the disease in the breast, DEXA, dual energy X-ray absorptiometry at the bone density, bone mineral density imaging techniques, and dental radiography, cephalometry, intraoral radiography, and pan tomography also there. Cone beam CTs are there. And up to this all comes under the diagnostic radiography, and PET, positron emission tomography, and single photon emission computer tomography. These are comes under the nuclear medicine. Here they are using the isotopes for the imaging purpose. And here that the magnetic resonance imaging and ultrasounds are also comes in the under the diagnostic radiology, even though they are not using the ionizing radiation. Magnetic resonance imaging using that strong magnetic field and radio frequency. Ultrasounds are using the ultrasound waves, that is between the 2 and to 20 megahertz ultrasounds are used. The frequency of an ultrasound can depend on the type of the scan and the desired image resolution. So furtherly, that diagnostic radiology can be divided into another 3 category. Already I spoke about the radio diagnosis. Another subdivision is that is called the interventional radiology, using imaging to image guide minimally invasive procedures like angiography, biopsies, stent placement. That all comes under the interventional radiography, which are involving with higher radiation doses. And here another one is radio oncology. High energy X-rays and gamma rays are used to treat the cancer. Cobalt 60 is LINAC is a best example for the radiotherapy instruments. And nuclear medicine also using that radiation. Isotopes or radiopharmaceuticals are injected into the body, and gamma cameras are used to detect the activities of the isotopes inside our body. So these are the major category of the radiology, and everywhere that protection is very important, and everyone working in that department has to use that radiation personal dosimetry. Why it is importance of radiation safety in radiology department? Because it induced health risk. It can cause cell damage, mutation, cancer, and radiation burns. And sudden high doses can lead to the radiation sickness, and chronic exposure, small amount of exposure continuously, if you are anybody getting, that can increase the risk of the cancer. So we are concerned about the protection to the patient first and the protection to the radiology staff. Protection to the patients is involved with the justification. Justification of the test is the priority of protection in the patients. Before prescribing any requisition for the test for the radiation involved test, there has to be analyzed the risk versus benefits. If the benefits are more, then we can go ahead with that kind of the testers. Otherwise, we can have to think about the alternative modalities like ultrasound or MRI scans. If the ultrasounds and MRI scans where that ionizing radiation is not involved, will give the, if you are supposed to give the good image quality or if the diagnosis, then it is better avoid to give the ionizing radiation to the patient. And another thing is, you have to follow the ALARA principle, that is as low as reasonably achievable, which means, and we have to give a minimum necessary radiation dose while achieving dye inverse accuracy. We don't want high quality images. We want a reasonable quality images that will be easily readable images that is more than sufficient to for the patients to give a minimum dosage. So, and the second thing is protection to the radiologist staff, which includes the personal protective equipment and personal radiation monitoring devices, and that has to be involved, because if the patients once in a while it comes to the department, but the radiologist stops, or it's continuously they are working in the radiation environment, so they has to follow certain protection rules and regulations formulated by Atomic Energy Regulatory Board. They have to wear the aprons, and then they have to use that personal dose monitoring devices during the but we see they have the personal protective equipment. So personal protective equipment, these kind of the lead aprons and lead masks are and gloves are used in the ancient period, the early 90s, and nowadays it's lightweight aprons and head caps and lead goggles and this kind of the thyroid shields and these lead gloves are used. And here, you can see that this kind of the protection is needed when we are very close to the radiation sources. If you are using the general radiography, this kind of the mobile protective barrier is more than enough to protect yourself from the scattering radiations. In general radiography, what will do that? The patient is lying in the X-ray table, and this is X-ray tube for the source of the X-ray. And when we are doing the exposure here from the radiographer is standing here and behind the mobile protector, it's lead shielded, or it can be a 9-inch wall with the lead glass. And when you are exposing, that the radiation will give scattering radiation comes from the patients. So if you are working in the general radiography, this kind of the protection is more than enough. We need not to wear the aprons or goggles, all the things. But you have to wear the personal monitoring devices. This slide shows how that interventional procedures are doing that. Here, that the doctors and supporting staffs everywhere, everybody, they are standing very close to the patient, and the X-ray source are here. They are standing very close to the source. That's why they are supposed to wear the lead aprons and goggles, all the things they have to wear, because they are very close to the radiation. So interventional procedures will give more amount of radiation to the radiology staffs. So personal radiation monitoring device has to be wear when we are working in the radiation field or in the environment, and to measure the exposure levels over time, ensure the safety and the compliance with the radiation dose limits. And the dose limits are stipulated by the Atomic Energy Regulatory Board, that separate film badges, thermal opinion dosimeters, and electronic dosimeters are nowadays available to measure the dose. So whom that this personal monitoring is necessary? It's a category described by the Atomic Energy Regulatory Board. Here that there are so many categories. Who are all working in the radiation field must wear the radiation monitoring device, and even these are the environment in the diagnostic radiology, interventional radiology, nuclear medicine, dental radiography. That these are the examples for the environment, and that their personal monitoring devices has to be used regularly. Why monitoring of the radiation is necessary? Because the radiation cannot be detected by the normal sense, and exposure to the radiation may induce biological effect in the exposed individuals. Therefore, monitoring devices like TLD badges are used to monitor radiation doses for occupational workers. Exposure to the radiation dose within the occupational limits produce no apparent indications of the biological effect. So whether we are within a limit or not has to be monitored with the help of the personal monitoring devices only. This in India, that's Atomic Energy Regulatory Board is the agency with monitoring the radiation facility and regulates the facility such as the X-ray facility by issuing license for the operation and of their X-rays equipment and approving designing of the diagnostic X-ray equipment and the layout of the working place. Here that ARB is prescribing the dose limits, and this limits has to be regularly monitored. And for the occupational exposure, for the workers, occupational radiation workers, this kind of the radiation limits are prescribed. So 20 millisievert must be per year. It should be within 20 millisievert. It is the radiation dose is measured in the unit of millisievert. So it must be less than 20 millisievert averaged over the 5 consecutive years, and it should not exceed 30 millisievert in a single year, and it should not exceed 10 millisievert in a single quarter of the year. And when it comes to the public, the patient, it should not cross the 1 millisievert. But the natural background is the 2 to 3 millisieverts per year. And for the lens, especially for because of the lens are very sensitive to the radiation, eye lenses are. So thyroid, breast, and gonads are the very sensitive to the radiation. So that's why that the lenses also frequently when we doctors or technologists are involved in the interventional radiology, the higher dose will be received by the eyes and lenses. That's why that lens, that 150 millisievert also is prescribed. That is the limit for the lens. That is separate dosimeters are available to measure the dose to the lenses, as well as the extremities also. You can wear that at the wrist level, at the finger level also. So for the skin, it is 500 millisievert, and for the extremities, that is the legs and hand, it is 500 millisievert in a year. For the public, it's at 15 millisievert, and it is comes to for in the case of the skin and extremities, it is 50 millisievert. For the whole body dose, and that personal monitoring device must be worn at the level of the chest level to calculate the whole body dose. It is measured in millisievert. So radiation detectors are used in the personal monitoring devices that can be classified into two major categories. One is active dosimeter, and it can provide the immediate reading of the exposure level on site. And the passive detectors are delayed reading dosimeter devices to measure the cumulative dose over the period of use. So nowadays we are using the thermoluminescent dosimeter, which are used to buy for 3 months period. Then only it's sent to the for reading purpose, and it will read and the data will be kept. That is comes under the passive detectors. These are some of the example for the active detectors. The gas-filled detectors and ionizing chambers, proportional counters, Geiger-Müller counters. These are the counters used with the gas for the ionization chambers. And these are the detectors are used for the survey meter to measure the radiations levels at the X-ray radiation vicinity. And these nowadays that the gas-filled detectors are grossly replaced by the solid-state detectors, scintillation detectors, and semiconductor detectors, which gives the real-time dose monitoring. And many active dosimeter includes alarms that alert the users when radiation levels becomes dangerously high, allowing immediate protective measures. So these are the active detectors are mainly used for the survey and the area monitoring, not for the personal monitoring devices. One of the active detectors only used in the personal monitoring, that is pocket dosimeter, that we've seen in the following slides. So passive detectors, these are used for the personal monitoring devices because it do not give a need to be monitored continuously, and for you can use it for some period, and then you can send it for the reading. And the film badge is involved in the first personal monitoring device. The film badge, it's 1950s it was introduced, and up to 1994 it was in use, and it is grossly replaced by the thermoluminescence dosimeter. Nowadays, this one we are using past 30 years, and some of the research labs and Atomic Energy Regulatory Board, they are using the OSL, optically stimulated luminance dosimeters, and in future it will be the TLD will be grossly replaced by the OSLD also. In European countries, they are using the RPLs, the radio photoluminance glass dosimeters, and India is not in use. In the TLDs we are using in India. What is the principle of the detection of the radiations? One is direct conversion. In the incident radiation, ionize the atoms, and which will it creates some mobile charges that are detected. That is called the ionizing chambers. Another thing is the indirect conversion in between the ionization and charge produced. It will be another conversion that's called the emission of the light. So it is an indirect conversion method, that the scintillation detectors or comes under the category. The signal charge is proportional to the energy of the observed. So how much is signals we received is directly proportional to the energy received. Let us see the one by one. It's a film batch. Film batch, actually this kind of the film batches we have used in the past years, and it is a very small and lightweight, inexpensive. We are in which it consists of film, dental film, like this dental film. This kind of the small dental film is used. It is a radio sensitive films, and it's a the radiation like a photography emission will be there. The photography emission will be affected by the radiation, and that all will be measured with the help of the densitometers. And some of the filters are available here. The 6 filters will be there, and it is to use the discrimination of the energy, and it can be measured gamma, X-rays, and beta exposures, and it will be that the film and all the things are placed into the holder, and it is about the 4.2, 3.5 centimeter in size. So there are 6 filters in the older film batch, and one is the open window, which records all kind of the rays, and the second one is the plastic window, which records the except all the radiation except alpha, because alpha will be filtered with the help of the plastics, and beta radiation. So and the third one is the cadmium, which will measure the neutrons, and fourth and fifth are the copper, thin copper and thick coppers. These are the thin copper is for the diagnostic X-rays, and thick copper for the therapeutic X-rays or the radiotherapy, oncology. And sixth one is the lead to measure the gamma radiation. How it works? It consists of the sensitive layer of this. This kind of the film is we are using. It's this is nothing but it's a very similar film using in the dental radiography, It is equipped with a thin cellulose acetate triacetate base or polyester base, which is coated with the silver halide crystals. When radiation falls on it, it forms a latent image. Latent image is nothing but the invisible image, and it will be become a visible image after the developing the film. Okay, that's a latent image is the image that's recorded in the film before the developing. And the radiation exposure causes ionization in the silver bromide crystals. So the silver bromide crystals are ionized and it is divided into the silver ions and the bromine ion and electrons liberated. And once all the silver ions are clumps in the sensitive areas of the film in the crystal surfaces, and after developing the film, this normal photochemical process is more than enough to develop this film, and each exposed grains are reduced to the metallic silver. During the developing, this metallic ions are getting the electrons from the developer and become into the metallic silver, and it is fixed. So silver undeveloped silver halide crystals are dissolved with the help of the fixer solution. So there will be a developer and fixer solution. So developing it will convert the latent image to the permanent image, and it will be fixed in the fixed solution. So at the end, you will get this kind of the film, which is a different density of the blackening of the on the film surface. So this degree of blackening is called the optical density. Okay, this optical density measured using the densitometers. So the amount of the radiation falls is directly proportional to the degree of blackening on the film. If it is very dark, that area, then higher radiation dose are received by the individual. If it is very light or it is very transparent, then less radiation is received by the individual. So it is purely based on the photochemical process and photography films used for the personal monitoring devices. The main advantage for the film badge is that it's lightweight, durable, and it's portable and cost efficient, because it's the film cost around 18 rupees only, and we need not to change the holders and the filters. Only we have to change and every month for the film only. That's how it cost around 18 rupees only. For the reading purpose, all the things, it will cost around 30 rupees. So less than 50 rupees, we can measure our personal dose, and it can discriminate between the X-rays and gamma rays and beta radiation. And for its a major advantage is the permanent exposure record. We can preserve it for a long period. If you want, we can retrieve it, and we can for any investigation interrogation purpose. The disadvantages can be affected by the heat and humidity, has to be preserved in a particular temperature, and sensitivity is decreased above the and below the 50 kilo electron volt. So the sensitivity is very less, and the advantage is it's a permanent exposure record. Now we move to the thermoluminescence dosimeter, which is nowadays we are using in our departments, and this kind of the thermoluminescence dosimeter we are using that. So it works based on the thermoluminescence. It's the process in which certain materials absorbs radiation energy and later release it as a light when heated. So when you are irradiating, it absorbs the energy, and when you heat the certain crystals, it will give the inner light form. So the light emitted is directly proportional to the amount of the X-ray energy stored over there. So lithium fluoride, lithium borate, and calcium fluorides are some of the chemicals used in Western countries. But in India, calcium sulfate is used because it is easily available in India. But calcium sulfate it has to be doped with dysprosium and other dysprosium for doping purpose. It is used to measure the external individual dose of from the X-ray, beta, and gamma radiation. It consists of the TLD card loaded in the cassette having a suitable metallic filters. So here the TLD card, this is the CSO for disker. So 3 discs will be there. The discs are sandwiched between the filters. These are the filters. It's the front side of the TLD dosimeter, And in the backside of the dosimeter, it's an open window. At the no filter, it's a perspex glass window, perspex window, and this is a copper aluminum window. And this is the card holder, and which having the CSO for disk. These are the three discs which was sandwiched between the two filters. A TLD core consists of the three CSO4 doped with the dysprosium and Teflon. And you can see a V cut over here. The V cut provides to ensure a fixed orientation of the card in the TLD cassette. And a, a thin paper wrapper which provides the personal data of the person using it. So these cards are actually it is around 13 millimeter diameter, and its thickness is 0.8 mm thickness. So it is fixed on the three holes in the card holders, and this card holder is wrapped with the papers which having the information about the individuals, and the specific number will be there. The number is the first four. These numbers are the institutional number, and the C, which means it is wear at the level of chest, so it will be C. If it is wear on the wrist level, it will be W. And this is the individual radiation workers in number, and this is the thing to where that period the TLD has to be used to protect all the things, and that's the way a plastic envelope is covered with that, and to avoid dust and mishandling. So TLD cassette look like this only. So there are three discs, a sandwich between the pair of the filters of the aluminum and copper. The top one is the aluminum copper filter. Behind this one, the copper aluminum filter will be there. Behind this, I will show in the next slides and the back side of the TLD cassettes. Behind this, the copper filter will be there. And second disc is sandwiched between the pair of the 1.5 mm thick plastic filter. This is the plastic filter, perspex filters. And third one is the open filter, open window. There is no filter here. So it will record all kind of the X-rays. This with the plastic will eliminate the alpha radiation. And this one, the aluminum and copper will record the gamma radiations, higher energy radiation. So it's look like this. It has to wear like this. When you open it, like it comes like this. It's the front page and back, and then there's the internal the CS4 disk with the cord. The back side of the front side. This is the front part, back side. This is the aluminum filter, perspective, and there's the open window. And here the back part, it's a copper filter is that. So TLD badges, it's a various kind of the types of the filters are available based on the our use. For general radiography, the chest badges are more, it's enough. And if you are frequently working with the radiation, very close to the radiation, or working in the interventional radiology like catheter cath lab or any radiology labs, then you have to work very close to the X-ray beam. Then wrist badges are available like this, and this kind of the finger levels of level small ring like thing is TLD badges are available. If you want to measure the dose to the eye, that's kind of the badges are available also. How it works, the CS over dysprosium when it exposed to radiation, how it works. You know that three energy bands, one the valence band and the in between the forbidden band and the conduction band. When exposed to the radiation, the ionization is happen, and the electrons are dislodged and free to move into the conduction band. So this is a valence band. When the crystals are irradiated, that the electrons are liberated, it will move to the conduction band. Then, while returning to the valence band, it is trapped to the sensitive centers in the forbidden zone. The forbidden zone is created by the doped agent like dysprosium, and when you reach ground level, then it is trapped in sensitive centers. When after exposing all the things, that when you are reading it, it is placed in the reader and the thermal energy is supplied. When you heat it, that to the passport by which the trapped electrons are released, and it goes close to the conduction band and it return to the ground level. So when you return to the ground level, the difference in the energy is released in the form of the visible light. The light emitted is directly proportional to the radiation dose absorbed by the passport. Here, X in the film batch, that's a blackening is directly proportional to the amount of the radiation absorbed. Here, the light emitted is directly proportional to the dose absorbed by the passport. This is summary. When exposed to the radiation, it goes to the conduction band and it's trapped into the sensitive center. And when you're heating it, it goes to the close to the conduction band, and when it returns to the ground level, when it emits an energy difference as a light. This light is absorbed with the help of the photomultiplier tubes. When you're heating, this is the TLD leader setup. Actually, that's it is the TLD crystals or CSO or discs are placed here. When you heat it, that light will emit, the light is sensed by the photomultiplier tube and it is amplified and it records the details with the electronic system. This is a typical TLD reader, looks like this, and a typical TLD batch readers can read up to 50 TLD cards at a time by loading them into the magazine. And this is the area, loading area. With the magazine, we have 50 TLD batches cards we can insert. That allows for the batch processing of the card without manual intervention between the each reads. But it is available in the some of the TLD authorized dealers only. So once you read it, you can get this kind of the wave thermoluminescence glow curve, and the curve look like this. It's a graph that shows the light sensitivity and temperature. So this related to the temperature to the light and light emitted intensities, and the peaks of the curve, it indicates the amount of the trapped energy released as a light, and which correspond to the radiation dose absorbed. The high peaks means that the high radiation exposure is involved. The advantage for the thermal TLD batch is the tissue coolant, and it's a better accuracy compared to the film batch, and not affected by the temperature and humidity like film batch, and it can be worn for up to 3 months, and a card can be reused. Once it is used, annihilation can be done. It erased all the data and it can be sent for the reuse also. This is a major advantage for the TLDs. And the limitations are no permanent. Once you read it, you erase it, then you cannot read it again and again. So another problem is that every time you have to calibrate it for the each batch. They told that the 50 TLD reader cards can be read at once, but before that you have to calibrate each and every time. Another in the future, the optical stimulated luminance is going to be replaced on the TLD readers. Because it is major difference is only the difference is the manner in which the electrons are freed from the traps. So all other things are same, and OSL dosimeters are more recent vintage than the thermoluminescent dosimeters, and here that very similar to the TLD, radiation absorption is happen. It's the trapped electrons are released by the optical stimulation. The TLD, we are using that heat energy to stimulate the trapped electron. Here the optical energy is used. The LEDs or laser lights are used to stimulation of the electrons, trapped electrons. And its advantage is it's easy, very faster, and multiple readout is possible. The most popular material used in the dosimetry is the aluminum oxide doped with the carbon. This OSLA typical batch looks like this. These are the 3 filters are there. One is open window, and this is a copper filter, this is a tin filter, and here the copper grid is also available to see whether it's active or dynamic radiation. All the things I can record it with this thing. In between this two filter front page, front side, back side. In between that, the layer of aluminum oxide doped with carbon is used in between, placed in between. It is used. It's very similar to the TLD badges. How it works? When aluminum oxide is exposed to the ionizing radiation, electrons are get trapped in the defects within the crystal structures. Later, when the material is exposed to the specific light source, that is LEDs or laser, the trapped electrons are released. And as the electrons return to the ground state and they emit the visible lights, which is measured using the photodetectors. So photodetectors are used here to detect the amount of the light emitter. The intensity of the light is directly proportional to the absorbed radiation dose. The advantages of OSL is it can be read multiple times. If you want to read, if you want any doubt, we can read it again and again. But when you read it second time, that at least 1% of the data loss will be there. And it can be used often. It can be stored for several years and no fading and with the external temperatures, no annealing required like TLD batches. And these also can be these batches and reused. The disadvantage is more expensive to when compared to the TLD. And it's essentially, but it is sensitive to the ambient lights. So it is when you are reading it, it needs a dark room, complete dark room. And only one pattern material, aluminum oxide, is nowadays available. Some other materials are available, but not in India. But in India, aluminum oxide with doped with carbon is available. But in research purpose only, it is used for routine personal monitoring devices. They are using the TLD only till date. And recent advance is RPL. RPL is a radiation monitoring device that measures the accumulated radiation dose using a special glass material that emits luminous light when exposed to the ultraviolet light after radiation. So here, that's almost the same thing, technology with TLD and OSL. Here that instead of the light, for the stimulation, ultraviolet light is used for the stimulation of the trapped electrons. It is widely used in the France and it is in the Japan also. A typical RPL dosimeters look like this. It's the front off and the back off. There's a buffers are there and three filters. You can see the three filters: tin, aluminum, copper, and open plastic windows are available, and open windows also available here. In between the two buffers, that separate glass coated with some silver atoms are used here, and the glass plate is inserted between the layers of buffer materials for protection. When it exposed to the radiation, the ionization is happening, and these glasses are doped with silver atoms. And when you're reading it, we're using the ultraviolet lattice with the 320 nanometers. And when you stimulate the electrons, you will get the luminance, and with the nanometers, it is absorbed with the fluorometers. This is the result of the RPL glass plates, and on the right side, this one is glass plate irradiated by the very penetrating cobalt 60 gamma with energy higher than the 1 millielectron volt. And irradiation appears to be uniform. You can see that the density looks like the color is very uniform because of the higher energy X-rays or gamma rays are penetrated all the filters, so it looks very uniform. What you can see in this one is irradiated by the low energy X-rays, and it is not penetrating and partially absorbed by the filters. That's why it looks like a different density. Here you can see the open area. It is different density. Other filters, the density is different, like this. So when you are using the violet and when you are off it, you look like this. This glass plates are. And when you are using the ultraviolet, it glows like this. The advantage of RPL is it can be read multiple times without losing data. So OSL always lose that data in the percentage of 1 and every time. But here that there is no losing of data. You can read it multiple times, and it's those information is permanently stored without any fading. We can use it for the permanent record as a permanent record, and we can investigate retrospectively. And it is suitable for both low and high dose measurements. And unlike TLD, RPL does not suffer from the signal loss over the time. Let us move into the final part of the pocket dosimeter. So pocket dosimeters are it's a passive active device, actually active device, and it's look like this, like a pin like this, and this is a charger. It has to charge before it used, and it's a very portable and radiation monitoring device that provides a immediate reading of accumulated radiation exposure. That is the advantage of the pocket dosimeters. So it is an active device used for the personal monitoring device. So particularly for the interventional radiology, where that higher doses involved. So it is divided. It is two versions. One is the analog version, look like this, and another one is the digital version. You can see the reading. The analog version, this scales will be there. You have to look on this for this reading purpose. And this kind of the chambers are used for charging the pocket dosimeters. Before the use, it has to be charged, and then only has to be used. It works on the principle of the ionization to measure the radiation exposure. It typically consists of the ionization chamber and an electrostatic system to detect the radiation level. So electrostatic systems are used, ionization chamber. You have to read like this, and this kind of the scale will be there. And this or fiber is the quartz fiber will be there. It will move around the scale based on the amount of the radiation ionization happen in the ionization chamber. So it is measured in the Röntgens, not in the millisievert. In later, it will be converted into the millisieverts by the calculations. It contains a small ionization chamber. This ionization chamber filled with the air, and there is a central wire anode and a metal quartz fiber. This is a metal quartz fiber, and it is positively charged and between the anode and quartz. And this is the eyepiece. From there, you have to see that it's a scale as fixed as it is a clip to insert into your pocket, and this is the objective lens, and this is the charging pin. So it has to be inserted into the chambers. When irradiating with the radiations, ionizations are happening, and these electrons are collected by and positively charged central anodes, and this collection electron reduce the net charge, positive charge, and allows the quartz fiber to return to the direction of the original position. So when it is before the use, it is deflected, and it is the end of the scale, and when you continuously using it, that's a charge. The electrons received in this ionization chamber neutralized, and the positive charge and the fiber quartz fiber return to the original position. It will be read like this. If you see that, I can see this quartz fiber scale and line and deflected. Actually, before the exposure, it should be in the 0. After the exposure, it will deflected towards the amount of the radiation based how much the radiation received by the individual. It is measured that the scales are which are measured in the milliröntgen. The major advantage for this instant reading, we can provide the real-time dose assessment, and it is very compact and portable. So no battery is required, because before the use we can charge it, and every time you have to charge it before the use. Reasonably accurate and very sensitive when compared to the other personal dosimeters. And limitations are it's fairly expensive. We cannot afford to each and every person working in the radiation field until it can discharge a due unit mechanical shock. If you are drop it, it will that reading will be changed, and it does not show the permanent record. At the end, the personal monitoring devices has to be, there are some rules and regulations, do's and don'ts, and should be wear at the chest levels, and it's a proper way. It's in a front side, back side has to be look on, and the proper way it has to be wear. And if any damage is there, and then it has to be reported to the radiation safety officer. Every institution has a radiation safety officer. That badges has to be replaced immediately. And TLD badge should be worn below the aprons, not over the apron. This is not correct. Below the apron it should be worn. Then only the actual radiation received by the individual is measured. And the TLD badges should be worn with the cassettes. Without cassettes should not be worn. The card only alone should not wear over that. It should wear with along the filters. It should be worn. And properly stored when you're not in radiation. So if you are working, coming to the department for the work, then you have to wear it, we take it from the storage and take it to for the wear it and work it. When you return to the duty from the duty, then it has to be replaced in a proper way. There is a separate place is there for the boxes there, but where that there is no radiation intervention area, that has to be placed in the proper way. Along with the TLD badges for the individual, a control badge is supplied from the service provider. A control badge, its a TLD card is used to, it is must be stored in a radiation-free area of the in the department, which measure the natural background radiation exposure, and which is then subtracted from the radiation workers, the TLD badge reading to determine the occupational radiation exposure. So natural background radiation alone recorded with the control badge, and the supplier will supply with all the individual TLD badges along with the control badge also. And some of the don'ts, and do not leave the dosimeter in the closed cars or in the sun or heat that can change the readings, and do not subject to dosimeter to wet or chemical environment, and do not take the dosimeter to your home. It should be placed in a particular place in a department where the control badge is placed. And do not intentionally expose to the dosimeter to radiation, or leave your dosimeter next to the source of the ionizing radiation. So it has to be should not be in the when not in use, should not be in the radiation close to the radiation source area, and do not lend your dosimeter to another individual also. Your dosimeter is for yourself only, should not lend to your friends. Do not use your dosimeter to monitor a personal medical processes such as your X-ray. For suppose somebody want to measure how much radiation is we're getting for a single chest X-ray, for that purpose it should not be used. It is used for the scattering radiation, to measure the scattering radiation absorbed by the individual working in the environment. And finally, thank you very much for the opportunity for the presentation, and my special thanks to Nitya Madam and Jaiselan sir for the opportunity. And if you want, I am having in my hand that's TLD badges. I will show you. This is the TLD, this initial film batch, what were used in our department in the early 90s. And there is a front page, front side, and there's a back side. You can see that 6 filters, the open, plastic, and this cadmium, thick copper, thin copper, and this lead. All the filters are there. There has to be, there is a clip over there. It is should be over there in the chest level, and we can open it like this. It look like this only. When you open it like this, it look like this only. This is the film. That's a dental film what you are using in our for the recording of the X-ray doses, and it has to be placed sandwiched between the all the 6 filters. It has to be fixed like this, locked like this, and it has to be weighed at the level of chest. And this one is a TLD, my TLD, and using for the current month up to March, it has to be used. There you can see that, and to open like this. It's a front part, it's a back part. You open like this. You can see that TLD cord. This is the TLD cord with the discus. These are the 3 discs. You can fix it here. You can see already that there is V-shaped cut. V-shaped cut is there. It's supposed to coincide with this projection. Aluminum filter, perspex filter. This open, and this is the rear side of the back thing. You can see the copper filter. This is the plastic, and this is the open. And when you are loading this, is must it that's a EV cut, and the projection must correlate, and has to fix like this. Should work at the level of the chest. That's all. And once in the 3 months, you can use it for the 3 months, and then you send it to the separate the authorized labs are available in Chennai. The Avantech lab is there. They measure the doses and will send the reports. Thank you once again for the opportunity.
Thank you so much, sir. Participants, do you have any queries? You can just ask Mr. Marimuthu, sir, regarding your doubts about the session. Dear participants, if you have any queries, and even though we can send sir's mail ID regarding the doubt, subjective matters, so that you can ask him a doubt and full clear explanation. Okay, okay.
Thank you, madam. Any clarification regarding this, you can contact anytime, and there is the email ID, and my phone number is with Vidya ma'am. You can contact anything, any research purpose. I'm ready to help, and we are doing so many research things. How much radiations are getting the patients for the particular studies also in our departments, and how much radiation is received by the individuals working in the department. These are the kind of the research are going on in our Institute of Radiodiagnosis at Kilpack Medical College Hospital. So if you want anything more details, you can contact me. And thank you for the opportunity.
I thank Mr. Marimuthu sir at this time, and I would like to tell you participants, like I'll be sharing the contact details of Mr. Marimuthu sir, so that you can just contact him further regarding any clarities needed for the radiational monitoring devices. And I would sincerely appreciate my thanks and gratefulness to Mr. Marimuthu sir on delivering a radiational personal monitoring devices. And sir, it is really an informative session, and knowing about the explanation of different types of radiations and machines, real-time equipments which are all related to radiation, those are all very informative and engaging, sir. And also, we learned about the new innovations which is done by you, like TLD and all. It's all really inspiring to know about the innovations by you and patented by you, and your expertise and clarity in presenting these complex topics made the session both enriching and thought provoking. And thank you so much for the wonderful session, sir. We are looking forward for much more sessions in the future also. Thank you
Sure,
so much for
madam.
the session once again. Thank you, sir. Thank you.
Thank you, madam. Thank you, madam.
Thank you, sir. Thank you so much, sir. Participants, you can just fill out the feedback link which is available in the chat box, and you can leave the session. Thank you so much for the wonderful support which has been provided by the day one session. Thank you, everybody. Good night. We will meet tomorrow at 6 o'clock for the next session. That is session number 3 on day 2. Thank you so much for your wonderful support. Thank you.